Intracellular calcium has been implied as an important part of several mechanisms of synaptic plasticity. These ideas can be tested by simultaneously monitoring changes in calcium and alterations in synaptic efficacy. Here we demonstrate how this is accomplished by combining calcium imaging with intracellular recording. Our experiments are conducted with a ganglion of the mollusc Aplysia californica. This preparation has a number of advantages, such as large identified neurons. The low temperatures that are natural for Aplysia slow signals and facilitate imaging. But the general techniques we demonstrate are easily transferred to other systems.
Hello, I'm Colin Evans in the laboratory of Elizabeth Cropper in the Fishberg Department of Neuroscience and the Friedman Brain Institute at the Mount Sinai School of Medicine in New York City. Today we are going to demonstrate simultaneous imaging of changes in intracellular calcium concentration and synaptic efficacy in the buccal ganglion of the mollusk Aplysia californica.
To detect intracellular calcium, we will inject a presynaptic neuron with the cell-impermeant calcium indicator dye, calcium orange. The preparation is then moved under a microscope and pre- and postsynaptic neurons are impaled with sharp electrodes. Repeated presynaptic stimulation results in an enhanced postsynaptic response, visible as increasing postsynaptic potential, or PSP, amplitude. Measuring the fluorescent signal of the calcium indicator dye will allow us to detect simultaneous changes in presynaptic intracellular calcium. To begin, anesthetize an adult animal weighing about